Today we're going to work on Unit 8, part of Bio 102, which is going to be the nervous and motor systems. So when you think about your nervous system, lots of times you might think of pain, you might think of electrical impulses that run through the body, but it's actually going to be all of that. So with this, we have a heritable sensory autonomic neuropathy. It's going to be a condition where people actually don't have this pain and they don't have feeling. See, here we have a little girl called Gabby Gringas, and she's actually going to feel no pain. Now with Gabby, she has a condition that is congenital insensitivity to pain and anhidrosis, which is actually going to be shortened into SEPA. And instead of being able to feel pain, she really doesn't have any feeling or sensation at all. So she doesn't feel hot, she wouldn't feel something cut her or if she was burned, or she wouldn't feel if somebody stuck a needle into her. So she wouldn't fear any type of shots. So most of the people who would come across this would think this would be a benefit, but actually it's not. Instead of being a benefit, this is a condition that fails to alert her that she's in a dangerous situation. She would not realize if she had a headache. She wouldn't realize if she broke a bone. She would not realize if she had a fever or had started to feel bad. So in a way, you might think it's a blessing, but actually her body wouldn't alert her that, hey, something's wrong. So the feeling of pain is actually going to be a necessary evil or a necessary part of being an animal and living. So this here is a very serious condition. Those who have this condition often have to constantly check themselves over and be very, very careful about what they do throughout the day. So what exactly is the nervous system? Well, it's going to be a network of cells that collects information about the organism's internal and external environments. It's going to process this information and it's going to send signals to effectors, then to muscles and glands that are going to be capable of responding to this information. You're going to have three main critical features of the nervous system. So the first one is that it's able to receive stimuli or a stimulus. With that, it's going to collect information about the organism's internal and external environment, like we said. So it can collect any information that it needs that it has to process to keep you safe. It takes that information it collects and it's going to process it. So it interprets incoming stimuli and then determines what type of response needs to happen. For example, if you accidentally lay your hand on a hot stove, it's going to send that information to the brain and the brain's going to interpret it and process what needs to be done. And so the third one is going to be an initiate response. So it sends a signal to the muscles and glands to respond to the internal and external environments there. So that would be, for example, maybe yanking your hand away. So inside this nervous system in your body, it's going to be made up of things such as the brain. You have your spinal column. You have your peripheral nervous system, which is going to come off. So basically, all of these are going to be made up of neurons. These are going to be the building blocks of your nervous system. They're going to be specialized cells that are going to be responsible for generating and conducting electrical impulses. And so with this, like we said, in all vertebrates, you're going to have your nervous system is going to be divided into two components. You have the peripheral nervous system and you have the central nervous system. Now with the peripheral nervous system, that's going to be all of the neurons and nerves that are going to come off that spinal cord. If it's part of the central nervous system, which you see up top, that's going to be composed of the brain and the spinal cord. And so those neurons and other supporting cells are going to help make up the brain there in that spinal cord. So with the peripheral nervous system, you have a network of sensory cells that receive information from the environment, and then the cells are going to transmit signals to effectors, and that's going to go to the organism's muscles and to the glands. So with the central nervous system, it's going to be made of the spinal cord and the brain. The central nervous system processes information that it's going to receive from sensory cells about the organism's surroundings, and it's going to send those instructions out to the nervous tissue on how to respond and act. So looking closer at this neuron, like we said, it's going to be specialized cells that are responsible for gathering and conducting electrical impulses. The building block of the nervous system is going to be what you see here. That's the neuron. And so with the neurons, it actually is going to be made up of different parts. Now, looking here, you have the main cell body, and then you're going to have the nerve fibers that are coming off. So with that cell body that you see there, it's going to be made up of the main section. It has a nucleus. That's where you're going to find all of your organelles. It's going to be basically like a generic cell or all other cells. It's going to be filled with cytoplasm. Like I said, the nucleus is going to be in the center. It still has all the DNA that's going to be there. And then you have your nerve fibers. So those are going to be things like the dendrites and like the axons. So with these nerve fibers, they're going to extend outward from the cell body, and they conduct nerve impulses. So here you see the dendrites actually picking up on the stimulus, and as it picks up on that stimulus, it is going to carry it through that nerve cell body, and then it's going to continue it on through the axon. So dendrites are going to be highly branched. They're designed to receive incoming signals. With the axons, they're designed to transmit information from the nerve to the next nerve or to an effector. What you see around it is going to be the glial cells. Those glial cells are going to be supporting cells. They're there to help protect, they insulate, and they nourish neurons. Now, if a neuron is slightly damaged, it can be fixed. It's when it is severed completely that it's not fixable. But if it has a little bit of damage, these neuroglial cells can go in and they can help repair it. Now with nerves, they're going to be comprised of neurons that are going to be bundled together with connective tissue. These structures are going to connect us to our world. They help enable us to see light. They enable us to hear sound, to touch, taste, and smell. Now, you're going to have let's go over the parts, the different parts. The cell body, which we just kind of went over, that's going to contain all the typical machinery of the eukaryotic cell. Like we said, it's going to have all the organelles, it has cytoplasm, and it has a nucleus. They're going to have two specialized structures that we went over. That dendrite is going to sense and respond to stimulation from outside of the cell. It sends information towards the cell body of the neuron. And then the axon is going to send information away from the cell body, and it's going to be a tube-like extension that comes off of the body of the neuron, and it transmits those signals that were picked up by the dendrites and the rest of the body's senses, and it's going to send those to be processed at the brain. Those glial cells, like we said, are going to be non-neuronal cells. They are just support staff, and so they're going to insulate, nourish, and protect. So going in, you're going to have three types of neurons. You're going to have sensory neurons, motor neurons, and what's referred to as interneurons. Now with the sensory neurons, they're going to collect information from the animal's environment. And so they're going to collect information from all five senses that you have. They're going to contain dendrites that are modified to respond to external stimulation. So for example, temperature, touch, taste, smell, light, and sound. Intraneurons, or let's go to motor neurons next. Motor neurons are going to initiate a reaction to a communication of stimuli. They help stimulate action by conveying signals to the muscles or to glands. And then you're going to have your interneurons. The interneurons are going to interpret signals coming from the sensory neurons, relaying them to the motor neurons. So they're going to be located in places like the brain and the spinal cord. So basically, here's an overview of what's happening when you pick up on a stimuli. So first thing, you're going to have your sensory neuron on your hand that's going to sense an irritation. In this case, it's going to be a mosquito that lands on your hands. And so it's going to send that signal to convey this information to your spinal cord. So it goes through, you see that purple section. That is going to go, that's your sensory neuron. It connects in with the interneuron. And so the spinal cord and the signal that was sent through the interneuron to your brain, that signal's interpreted and a response is determined. This is that you want that mosquito to go away. And so then you're going to send that information back down through an interneuron, and it's going to connect into a motor neuron. That signal that connects into the motor neuron is going to have you swat that mosquito away. Some signals are going to be able to bypass the central nervous system and travel directly to the sensory neurons, to the spinal cord, where they're going to connect directly with motor neurons. These are going to be called reflexes. So these are basically going to be an automatic response. Like we said, it occurs when the signal travels directly from the sensory neuron to the spinal cord and connects with the motor neuron. It's going to stimulate a response to a sensation that does not need the brain to process it. And so here it's going to have two functions. The first function for reflexes is that it's going to help you maintain your homeostasis. So this is going to be important. It helps maintain your heart rate. If you need blood pushed out to the body in different areas a lot quicker, it's going to speed up that heart rate. Or if you're sitting around, relaxing, getting ready to go to sleep, it might reduce that heart rate. It also helps with the respiratory rate. It helps with your blood pressure. Another thing that it's going to do, another function, is it provides protection. So it could be a withdrawal reflex, it could be sneezing, coughing, vomiting, it could be swallowing as a reflex. So these are all going to be things that are going to help protect you. We talked about the withdrawal reflex, like when you laid your hand on something hot, like a hot stove. Automatically, it does not need the brain to process it. You can yank your hand away because that reflex will take care of it. Now think about this. The speed of my reflex is not affected by distractions. For example, driving while talking on a cell phone. What do you think? Well, they've actually found that if you're in a deep conversation, it could cause a difference in your reflexes or your reaction. And they've noticed that it actually affects it a lot more as you get older. So how exactly do these neurons work? Well, we have dendrites. They're going to be the receivers of external stimuli. And it's going to occur in two ways. You're going to have motor neurons and interneurons that are generally going to connect and receive signals from other neurons. And then you have sensory neuron dendrites, on the other hand, that are going to be modified to respond to specific external stimulus such as a touch or a sound, light, or even maybe chemicals. So the dendrite in action. So the first thing that's going to happen is you have the receptor proteins within the cell membrane of the dendrite that are going to respond to stimuli by briefly opening up little chemical channels. And you see those there in purple. These are going to be your ion channels. These open channels are going to allow the passage of ions, momentarily altering the electrical charge within the cell from the negative charge it carries prior to the stimulation, where it's going to be at rest. Third thing that's going to happen is the signals from all of the dendrites of the neuron coverage. If the sum total of signals coming in significantly positive, then the cell initiates an action potential that travels down the axon. And so you see the action potential coming in and traveling down there in the green. So action potential is basically going to happen at the body of the cell. And so what happens is that cell body is going to integrate it. So if the sum of total signals coming in is going to be positive, then the neuron's going to initiate an action potential, and an electrical signal is then going to travel down that axon. So what happens is, like we said, it occurs. The dendrites are going to receive that stimuli. It's going to come in, and it's going to change over to an electrical signal that travels down the axon. So action potential propagation is going to occur when the action potential moves down the axon, and like we said, it's going to cause sodium ions to rush into the axon, and it changes that cell's negative charge to positive. So once it reaches what's known as terminal buttons, the action potential is going to initiate the release of chemicals, and then it's going to allow that body to, once it hits those terminal buttons, It's going to act like a knob at the ends, and it's going to allow the axon to close around that muscle section and around the glands or the dendrites, so it can send that signal across. That's going to allow the body to regulate its nervous impulses. It's going to allow it to work efficiently, and it's going to allow it to control what it needs to control without you having to sit there and think about it. So with graded sensations, this is going to be the intensity of the sensation an individual feels. It's going to be modulated by the number of neurons that are going to fire at a time. So however many are going to be stimulated at the same time. So here you see this guy. He is holding two different balls. One's a dodgeball and one's going to be a bowling ball. And so the intensity of the sensation is going to be modulated by the number of action potentials per unit of time and the number of neurons that are stimulated. And so with that dodgeball, it's not going to need to stimulate as many as the bowling ball because it's going to be much heavier. And so here we have, in a response to action potential, the axon terminals are going to release those contents of the vesicles into the extracellular matrix and potentially influence those adjacent cells. So looking here, we see a picture of the axon as it's coming down. And so it was picked up by the dendrites, remember, and it's going to travel down the axon, and you see those terminal buttons that I was talking about. Notice that it has a nice myelin sheath. That myelin sheath is going to be created by what's known as Schwann cells. These cells are almost like a jelly-like substance that wrap around and grow around the axon. As the action potential travels down the axon, it will speed up once it hits those myelin sheaths. The in-between section, where it does not have a myelin sheath, it's going to slow down a little bit. And so here in this picture, you see how the action potential is actually moving down that axon. So at those gaps of the myelin sheath, it's going to have ion channels that allow sodium ions, which carry that positive charge, to rush in. And it's going to change the cell's charge to positive in that region. And thus it's going to propagate the action potential down. So in those regions, it does travel a little bit slower, and those are called the nodes of Ranvier. And so this just shows you some myelin sheaths, again, that are going to be wrapped around this axon at the bottom of the page. Basically, it's a type of insulation. It's going to be a fatty coating. Like we said, it is created by the Schwann cell, and it prevents the action potential from weakening as it travels down the axon. So it helps it travel fast. And so with this, it is going to be kind of a white color to it. Areas of your brain are going to be rich in axons that appear white, and that's going to be regions that are going to contain a lot of this myelin there. And so brain regions that are rich in cell bodies and dendrites appear to be gray. If you have a lack of myelin, it's going to be a lack of myelin on those axons. It can be seen in babies because it hasn't quite developed yet. And so when they first start to walk, you'll notice how they don't have a lot of control and how it takes them a while before the myelin develops around their axons before they can actually walk properly. So at the time, the myelin hasn't completely formed, like we said, and gross motor control isn't very good. At the synapse, neurons are going to interact with other cells. So this is going to be the end of an axon. That axon's terminal or terminal button's always going to be right next to another neuron, or it's going to be next to a muscle cell or a gland. And so that is going to be the point where it comes together. And that point where it comes together is referred to as a synapse. So when I say synapse, it's just the junction between two neurons. It could be the junction between a neuron and an effector. And so it's going to have 7 main components. It's going to have sacs that are called vesicles. They release neurotransmitters into the synaptic cleft. It's going to have a neurotransmitter that's going to diffuse to the nearby receptor sites. It's going to have a neurotransmitter that's then going to attach to the postsynaptic receptor. The gate is then going to open in the postsynaptic cell membrane. Once the gate's open, it's going to allow the signal to pass to the postsynaptic cell. Then that neurotransmitter is going to be released from the postsynaptic cell's receptors, and the neurotransmitter is going to be recycled or broken down. And so that's what's going to occur. So notice here that axon terminal is going to be at the end of the neuron. It's going to have that calcium channel there. It's going to have the terminal button, which you see is going to be at the end. You see all the synaptic vesicles that are going to be there. And then you see where the neurotransmitter will come in. So let's start off. So with step 1, you see that action potential is going to reach the axon terminal of the neurotransmitter. It's going to be filled in those vesicles. That neurotransmitter is actually, if it's going to be with a muscle, it's called acetylcholine. So we'll use that one for example. So let's say all those little vesicles are filled with acetylcholine. They're going to come across and they're going to attach to those purple receptors. When they attach to the purple receptors, it's going to open up those channels. Before, the ions cannot cross over. So they stay in that synaptic cleft, which is going to be the opening between that little button there, the axon terminal, and the cell that's receiving the information, which is in the yellow there. And so once it opens it up, it's going to allow those positive ions to travel across into that cell. Once they travel across, then the neurotransmitter molecule is then going to be broken down and can be reused again and put back into the vesicles of the cell that it just, or the end it just came from. So like call screening for your brain, your body's going to be able to use this, and they're going to be able to block some information that's coming across because you don't necessarily need all information that's coming across. So this option is not just useful, but it's essential to your nervous system. It helps you to control what comes through, what doesn't. Could you imagine taking in all the information throughout the day? It would literally probably drive you crazy. So continual stimulation, that's going to be when most neurons gradually reduce the amount of neurotransmitter that they release, and thus it's going to reduce the strength of the signal that's coming across. There are going to be many types of neurotransmitters. There's going to be about 25 total. I gave you one when I showed you that example. I talked about acetylcholine. But like I said, there's going to be about 25 that we've identified so far. And they all have specific, like a personality of how they're going to act and what they're going to do. So some important ones that you need to know, we talked about acetylcholine, but you can also have glutamate, dopamine, and serotonin. So acetylcholine, often referred to as the neurotransmitter used for muscles, is going to be released by motor neurons at the point where they synapse with muscle cells. When enough acetylcholine binds to a muscle cell, that muscle cell is then going to contract. Curare is going to work by blocking a receptor site where acetylcholine binds to muscle cells. So once it gets into the animal system, that curare disease is going to create a form of asphyxiation and anaphylaxis, basically, because it's going to make it impossible for the skeletal muscles to contract. And so here we have a hunter that's collecting shavings of a plant that's going to actually be where they get this curare and extract it. And they're actually going to use it as a lethal poison that they put on darts or that they use to actually go hunting with. Glutamate is going to be a neurotransmitter that's an excitatory neurotransmitter. It's going to appear to be involved with things like learning and memory. Dopamine, I'm sure everybody's heard of this one. This is going to be an important neurotransmitter that initiates and coordinates movement. It's also one of the body's chief happiness neurotransmitters. And then serotonin is generally going to function as an inhibitory neurotransmitter. It helps affect things like appetite, sleep, anxiety, and mood, and it helps produce feelings of contentment and satiation when it's going to be released. Sensory receptors are going to be our windows to the world around us. So sensory equipment is going to be located at the front of bilaterally symmetrical animals. And that's going to make sense because it's the first part of the animal that's going to encounter new things in its environment. So with these senses, you have 5 of them that the body's going to use. You have your hearing, touch, taste, smell, and sight. These are going to help make you aware of your environment around you, and they're all basically going to work the same. And so with this, they're going to work with one of 2 ways. So your dendrite on a sensory neuron is going to be stimulated, and that sensory neuron is going to do one of these 2 things. It's either going to fire an action potential, which means it's going to basically shoot down the axon and reach part of the brain where the signal is going to be perceived, or it's going to alter the rate of a neurotransmitter being secreted. And so basically what that means, it increases or decreases the rate of action potential firing into that neighboring cell. So with that, the amount of stimulus basically is going to determine the sensation intensity. The sensation, basically, the intensity is going to be determined by the number of action potentials per unit of time and number of neurons that you have stimulated. So if it's going to be a strong stimulus, that means you're going to have a higher sensation of intensity like we saw with the balls. Or if it's a weaker stimulus like we saw maybe with the dodgeball, you're going to have a lower sensation of intensity. So going into our 5 senses, first we're going to start off with taste. Taste is going to be detected by taste buds that are going to be found on the tongue. There's actually going to be more than 10,000 taste buds there. It's going to involve things known as chemoreceptors. The chemoreceptors are going to have about 60 to 80 cells or sensory cells that are going to be found within each taste bud. And in order to taste, it's going to be part of a 3-step process. So looking here, you just see basically the outline. You have your tongue, you zoom in, you see those sensory neurons. And then with the sensory neurons, you can see how the action potential is going to be sent down through a axon to the brain. And you see the receptors, just like in the others. So particular taste receptors are going to allow only specific food molecules with exactly the right shape to bind to them. The 5 groups that you're going to have are going to be sweet, salty, sour, bitter, and umami. And umami is going to be one that you might not have heard of a lot, but that one's going to be a savory type food. And so with these, what's going to happen is that it's going to travel down and basically occur in 3 steps. You're going to have it stimulated with particular chemicals in food that get dissolved in the saliva. And those dissolved chemicals are going to bind to your taste receptors on cell surfaces. And so not all animals are going to sense and taste in the same way. For example, some insects have chemoreceptors on their legs. They'll be able to taste on their legs, and others are going to have taste receptors on their antennae or tentacles. And so like we said, those chemicals are going to bind to your taste receptors on the cell surface, and only specific ones that are a specific shape are actually going to make it through. This shape is going to allow specific molecules to fool the brain. So if you have any type of artificial sweetener, the molecule is not actually going to be sugar, but because it closely resembles sugar in its shape, it can stimulate the same taste bud receptors, and then the brain's going to perceive it as sugar. Smell receptors are going to be in the nose, and they help detect airborne chemicals. So our senses of smell are going to work in almost exactly the same way as our sense of taste. Airborne chemicals are going to move through the mucus in the nasal cavity, and they're going to bind to smell receptors. That's going to trigger action potentials that shoot down the axon all the way to the smell center of the brain, where then that signal is going to be perceived as a particular odor. And so here we have smell. It's going to involve, like we said, the chemoreceptors. Airborne chemicals are going to move through the mucus into the nasal cavity that you see. The chemicals are going to bind to the smell receptors on that cell surface. And so you see those chemoreceptors. And then chemical binding is then going to trigger an action potential that delivers the smell to the brain. Now, Smell can be slightly different in other animals. For example, some insects are going to have smell receptors on their antenna. Some reptiles are going to have smell receptors on their tongue. Human females are significantly better, for example, than males at detecting, distinguishing, and identifying odors. Even more sensitive during ovulation, actually. So you can even pick up on more smells if the female's ovulating. And so here we have dogs picking up. So dogs are going to have as many as 40 times more receptors than a human would. However, they're going to have less taste receptors. So they don't taste as well as humans, but they smell 40 times better. Or they can pick up on smell 40 times better. Next, we're going to move into vision. And this is going to involve photoreceptors. So despite the great diversity of species with light sensing capabilities, the basic functioning of light-absorbing cells is going to be quite consistent. So the function of these cells can be broken down into 5 steps. So here with vision, we have dark vision. So basically what happens here, sodium channels located within the photoreceptor cell membranes are going to open up. They allow the photoreceptor cells to continuously release inhibitory neurotransmitters. The inhibitory transmitter is going to reduce the ability of adjacent neurons to excite the neurons that signal the brain. And an action potential is not sent through the optic nerve, and light is not going to be perceived by the brain. In light is going to cause the light-sensitive molecules to change the shape. It's going to close the sodium channels in the photoreceptor cell membrane. The closing of the sodium channels is going to reduce the amount of inhibitory neurotransmitter that's released by those photoreceptor cells. No longer inhibited, the adjacent neurons are going to be able to excite and send a signal to the brain. And then an action potential is going to be sent through the optic nerve, and light's going to be perceived by the brain. There's going to be 3 different types of eyes that have evolved over time. First, you have what's referred to as the eye cup. It's going to contain photoreceptor cells that detect presence and intensity of light. They can't form images though, and they can only basically tell if there's going to be light around. And some examples of this is what you see in the picture. It's going to be a flatworm. Next, we have the compound eye. It's going to contain many separate light sensing units with its own lens. These units are going to detect light onto a dozen photoreceptor cells. The brain's then going to interpret those signals as an image. It's going to help enable the insect to see different wavelengths that aren't visible to us. And examples of this would be things like the fruit fly you see here. And next, you're going to have a single lens eye. That's going to be like what we have as humans. So with the single lens eye, it's going to contain a single lens that helps focus that light into a highly sensitive photoreceptor cell. It's going to consist of some main basic components. And we'll look at those in just a little bit. So with these compound eyes, it's going to be made up of separate or multiple separate light sensing units. So we have what's known as the iris, we have the retina, and in the retina we're going to have rods and cone cells that are going to be in there. And so looking here, you see some flowers that are going to have ultraviolet light reflecting petals on them. We cannot pick up on this, but insects can. Going into the single lens eye, you see how you have the iris. That's where the light's going to enter through. It's going to be the colored portion of the vertebrate's eye. It has an open section, which is referred to as the pupil. And so that opening is going to control how much light actually goes in and out of the interior part of the eye. Next, you have the retina. That's going to be found on the inner surface of the vertebrate eye. It's going to be composed of basically a sheet of nervous tissue. And that nervous tissue is going to have light-sensitive cells on it. It's going to help transmit impulses to the brain, which are going to be picked up in the vision center. You're going to have 2 types of these photosensitive cells that are going to be found on the retina. One type is going to be a rod. It's a highly sensitive cell is going to pick up on tiny amounts of light, and it allows vision at night and when it's dim out or it's in a low light area. Cones are going to be less sensitive to light, but they're going to allow vision during daytime, and they actually help detect color. There's going to be 3 types of cones. You have your red sensitive cones, blue sensitive, and then green sensitive. Eye location is going to be important. So the farther apart the eyes are, the wider the animal's field of vision is going to be. And so the wider the field of vision is going to allow animals to see what's around them, and it helps aid in detecting potential predators. Eyes that are going to be closer together are going to actually reduce the field of vision that allows for accurate focusing, and that's going to help aid in things like hunting. Going into our next sense, we're going into hearing. So it's going to be basically sound waves being collected by the ears, and then it's going to stimulate an auditory nerve. So it involves what's known as mechanoreceptors. These are going to be specialized neurons with receptors that respond to mechanical pressure. So basically, you're going to have sound waves that are going to create tiny fluctuations in air pressure. That's going to be collected and amplified by the ear, and then it's going to pass that information onto the brain. So here in these steps, first you have your sound waves that are going to be funneled down into the ear canal. Then the sound waves are going to cause the eardrum to vibrate. Those vibrations are going to be passed on to the inner ear membrane. Fluid in the inner ear is going to conduct the vibration, bending the hairs that are shaped receptor cells, and it's going to release a neurotransmitter. That neurotransmitter is then going to cause the auditory neurons to fire an action potential, and that signal is going to be then passed to the brain where it's interpreted as sound. Hearing loss is going to occur when the inner ear hair cells are going to be damaged. It's going to reduce the ability to stimulate auditory neurons. Once that you damage those cells, they do not repair themselves, so hearing loss will be permanent usually. Insects can hear with their antenna hairs. They don't have ears per se. So insects can also generate communication by sound, by beating their wings, or by rubbing their wings together. Some animals are going to use things like echolocation. And so echolocation is going to be a sensitive call and response system that certain animals such as dolphins or bats will use. They're going to do this by emitting high-pitched squeaks and then wait to hear the sound as it bounces off the surface and comes back to them. Touch is going to be the brain perceiving pressure, temperature, and pain. So touch is actually going to be a class of sensations that's generated by numerous different types of sensory neurons that are going to be sensitive to things like pressure. That's going to be your mechanoreceptors, temperature, which are going to be your thermoreceptors, and then pain, which are going to be the pain receptors. So when you're doing the sense of touch, you can see how there you're going to have a pin sticking into the hand. The mechanoreceptor picks up that, hey, there's pressure. The pain receptor is going to pick up, hey, it's actually gone in and damaged the skin. And then the thermoreceptor might pick up on the air or the temperature around it. Phantom pain is actually going to be something that most amputees report that they sometimes experience. It's going to be a pain that comes from where their amputated limb once was. And so with these sensations, they are going to be real, and they occur when the neurons in the brain that used to represent touch receptors in that limb start to fire. So basically the brain is firing mis-signaling or shouldn't be firing at all. Other senses that are going to help animals negotiate the world are going to be things such as balance and motion. They have electricity. We're going to have magnetism, and we have heat. So with the balance and motion, it's going to involve cells being sensitive to gravity, velocity, and orientation. So animals that will use this are going to be things such as jellyfish, lobster, crayfish, and even humans. We have our magnetism, It's going to involve a wide range of species, from birds to eels and sharks, migrating beluga whales, and even some bacteria are able to detect the Earth's magnetic field, and it's going to help it to navigate throughout the world. If an animal is able to see heat, it's going to involve cells that are going to be sensitive to heat that's found in pit organs. These are going to be organs that are used to see heat differences in the environment. And so you can see this in animals such as pythons and vipers. And we didn't talk about the electricity. We didn't have a slide for that, but that's going to involve things such as cell sensations. It'll be sensitive to different electrical charges. Remember that your nerve impulses are going to be a type of electrical charge. And so some animals that are able to use this and help it to track down its prey are going to be sharks. Now going into the muscular and skeletal system, it's going to help you be able to move. There are going to be 3 types of muscle tissue. You're going to have skeletal muscle, cardiac muscle, and smooth muscle. Now with skeletal muscle, this is going to be muscle that you control. So your nervous system will help control this, and you can actually think about what you want to do and make yourself do it. The reason it's called skeletal muscle is because it's usually going to be attached to part of the skeletal system. And so here you're going to see 2 muscles that are working together in opposition, so it can contract one muscle and move it in one direction, while another muscle is kind of relaxing to help move it in, help it go, and then you can contract the other muscle to help it move it in a different direction. And so with skeletal muscle, it's going to be made up of special muscle fibers, and those muscle fibers are actual muscle cells. So let's go back to skeletal muscle for a minute. Like we said, it is controlled by the person, so it's going to be voluntary. It's responsible for generating movement, and it attaches to bone by a type of tissue called a tendon. And so that tendon actually attaches onto the muscle, and then part of it will go in and attach to the bone. It's going to attach to muscles at 2 sections. So insertions are going to be the movable end of the skeletal muscle, and then the origin is going to be the immovable end of the skeletal muscle. And so it's going to be controlled by individual neurons that are attached to each muscle fiber. Like we said, a muscle fiber is simply going to be a muscle cell. And it's going to make up about 40% of the human body weight. So your skeletal muscle is going to be 40% of your weight. Humans are going to have around 700 skeletal muscles. If you eat meat a lot, that's actually going to be the skeletal muscle of that animal. So going into our muscle vocabulary a little bit, we said that the muscle fiber is simply going to be a muscle cell, but with multiple nuclei and numerous myofibrils. And so the myofibrils are going to be thread-like structures that are going to have contractile units in it, and they're going to shorten when they contract. These contractile units are going to be made up of 2 types of protein. These proteins are actin and myosin. Actin is going to be a thin filament, and myosin is going to be a thick filament. And so they're going to work together to attach to each other to contract and to shorten that muscle to cause movement. And so looking here, you can see that you have 4 different steps to contracting a muscle. First, you're going to have what's referred to as the detach. That's where a link between a myosin and an actin filament, which are parallel are going to be broken, and a molecule of ATP is going to bind to that myosin. So that myosin is going to be the thick filament that has the little heads that stick up. The second step is, as the ATP breaks down, energy is going to be released, and it alters the shape of the myosin head into a higher energy shape. Third step is going to reattach. And so that altered shape at that myosin head is going to reach further down the actin filament where it's going to attach to it. Fourth step, it's going to pull back. So the myosin head, which is attached to the actin, is then going to snap back into its original shape and is going to pull that actin filament as it does this and shorten that fiber. So here we have what's called the fast twitch versus the slow twitch. So when a muscle contracts, the duration between the contraction and the relaxation is going to be called a twitch. There's going to be a slow twitch and a high twitch fiber that you can find inside of muscles. World-class sprinters are going to be known for having more fast twitch fibers, which enlarge in response to heavy use in the leg muscles, while world-class long-distance runners are going to have more slow twitch fibers in their leg muscles. So going in with, let's finish up with the muscles. When we looked at these different units, that sarcomere is going to be referred to as a contractile unit. It's going to be a fundamental unit of that myofibril. It consists of long overlapping filaments. Like we said, it's going to have two types. You're going to have your actin and your myosin. The thin filament is the actin, and the thick filament is going to be your myosin. With the skeletal system, it's going to have 5 functions. It's going to have structural support, which is going to give shape and structure, including securing organs in place. Protection, which is going to shield vulnerable tissues such as the brain and the heart from external insults. It enables movement, so it attaches with muscles, and where it connects, it can help generate movement. Cell production is going to be responsible for making red blood cells, white blood cells, and platelets, along with plasma. And then it acts as a mineral reservoir. So elements such as calcium and phosphorus are going to be stored in the bones in response to deficiencies and excess in the bloodstream. So if you have a deficiency, it's going to go in and eat away and release those minerals into the bloodstream. If you have an excess, it's going to go in and it's going to put those onto the fibers of collagen that support or start to make up the bone. And once it deposits those, it's going to make the bone thicker. So there's going to be different types of skeletal systems. You have the hydrostatic skeletal system, which is going to be a fluid-filled cavity with it's going to have a sufficient rigidity, and it's going to have muscles that surround it that help it generate movement. This is going to be found in things like jellyfish that you see here, but you can also find it in earthworms and squid. You also have an exoskeleton that's going to be a rigid outer covering that supports, protects different types of animals. It's going to help reduce desiccation, which is drying out. It may consist of calcium, or it could be made up of chitin. It's going to be found in things like turtles, mollusks, and insects. And then you see at the bottom, you're going to have an endoskeleton. This is going to be an internal support structure. It's usually composed of a hard mineralized tissue, and there's going to be 3 different types of tissues here. You have the calcium-based tissue, which is going to be similar to bone. You have cartilage, and then you have actual bone. So vertebrate skeletons are going to have 2 divisions. You have what's referred to as the axial and the appendicular skeleton. And so with the axial skeleton, that's going to be made up of the skull, sternum, ribs, and vertebrae. With the appendicular skeleton, that's going to make up your limbs, your pelvic girdle, and your pectoral girdle. If the bone starts to break down, that could lead to what's known in humans as osteoporosis. This is going to be a disease that occurs as bone density decreases and chemical composition of the bone changes. It occurs most commonly in women, and for 2 reasons. Women's bones are going to be less massive than men's, so less bone mass, so it's going to be a lot smaller already. And the production of estrogens in women start to decline dramatically in menopause. And so that's going to actually start the breakdown of that bone. Some different risk factors for osteoporosis would be a family history. If you're of European or Asian ancestry, you tend to have higher chances. Thin or small body build tends to have a higher chance. Inactive lifestyle, so if you're sedentary and you don't move around a lot. If you smoke cigarettes, it'll break down your bone a lot faster. A diet low in calcium and vitamin D, more than 2 alcoholic drinks a day, and use of certain medications or treatments can break down your bone. Things that you can do to help improve are going to be take a vitamin D supplement, have a diet that's going to be high in calcium or vitamin D, perform regular weightlifting or weight-bearing exercises, and medications. Next, we're going to cover the brain. The brain's going to be the largest and most complex portion of the nervous system. It's going to be composed of 100 billion neurons. It encounters nerve centers associated with sensory functions. It's going to be responsible for sensations and perceptions. It's going to issue motor commands of skeletal muscles. It's going to carry on high mental functions like memory and reasoning. It's going to contain centers for coordination and muscle movement. It contains centers and nerve pathways that are going to regulate visceral activities, and it provides characteristics such as personality and things like that. It's going to be made up of 3 different brain regions. You're going to have the hindbrain, the midbrain, and the forebrain. And so looking here, let's start out with that forebrain. The forebrain is going to have a cerebral cortex that you see there that's going to be involved in abstract thought, problem solving, and language. It's going to be the largest region, the forebrain is, that's responsible for most of what we consider to be the higher thought. It's going to help determine things such as perception, memory, language, intelligence. It's what helps put together your personality. So with this forebrain, you have that cerebral cortex, which we had started talking about. It's going to be the outer functional portion of the cerebrum. It's responsible for all conscious behavior. It's going to make up about 80 to 85% of the brain mass in humans. It consists of about 10 billion neurons and hundreds of billions of synapses. And so it actually has those 4 main regions. It's going to have a frontal lobe, it's going to have a temporal lobe, a parietal lobe, and an occipital lobe. Next, we'll go to the thalamus and the hypothalamus. That thalamus is going to be one of the primary switchboards of the brain. It receives most visual and auditory and touch input. It's going to help relay signals to the cerebral cortex. The hypothalamus is going to be your chief regulatory center of the brain. It helps to regulate things like hunger and thirst, sexual activity, body temperature, hormone secretions. It's going to contain pleasure centers, so it provides mechanisms by which animals can be motivated to engage in behaviors that are going to be essential for survival as well as reproduction. Now, going into the different types of lobes with the cerebral cortex, we had what we called the frontal lobe. That frontal lobe is going to regulate things like speech production, motor control, smell, problem solving, personality. And that's going to be in the front portion of the head. The temporal lobe is going to be where the temporal bone is. That's going to be on each side. That's going to perceive and process auditory and visual sensations. It's going to be important in pattern recognition and language comprehension. You have the parietal lobe, which is going to be the back section of the cerebral cortex. That's going to receive and perceive touch and pressure sensations. It's also going to be important in sensory integration. Sorry, parietal's on top. Then the occipital, that hit me at the end, the occipital lobe is going to be the back, and that's going to receive and process visual information. Next, you have your midbrain. Now with the midbrain, that's going to be important in filtering and evaluating motor and sensory neurons there. So with the midbrain, it's part of the brain stem. It's going to help filter, like we said, and evaluate what's coming in and what leaves that brain. And so if you look at it, it's going to be the actual top part of the brain stem. Then you go into the hindbrain. That hindbrain is going to be made up of the cerebellum, the pons, and the medulla. The hindbrain is going to be located at the top of the spinal cord. It's the beginning of the brain. It contains three main areas, like we said. So the cerebellum is going to be responsible for coordinating motor activity. The pons is going to be a pathway for the motor and sensory neuron signals. And then the medulla is going to be a pathway for motor and sensory neuron signals. And it's going to help regulate basic physiological functions such as respiration, heart rate, and digestion. So going into the hindbrain a little bit, we said that was at the top of the spinal cord. It's the beginning of the brain, and it's where the spinal cord expands into the hindbrain. We talked about the three main parts. With that midbrain, it's going to be along with the medulla, the pons, the hindbrain. It makes up what's known as the brain stem when you go into the brain. It's going to help filter and evaluate the importance of each signal. And then you had the forebrain. In humans, the forebrain is going to be the largest region of the brain. It's going to have two control and relay structures, the thalamus and the hypothalamus, as well as the cerebrum. It's going to be responsible for most of what we consider to be our higher thought, higher thinking. It helps with our perception, memory, language, intelligence, and your personality. Now with that pleasure center, in the 1950s, James Olds inserted a tiny electrode into the hypothalamus of the rat's brain. And so here he's going to study that pleasure center. When he excites it or when he sends a signal to the rat's brain, it seemed to experience great pleasure. And so this is how he discovered that this is going to be the pleasure center of the brain. The cerebral cortex and Phineas Gage. The cerebral cortex is going to be the most sophisticated part of the brain. The guy that you see here in the picture is going to be Phineas Gage. He's going to suffer from severe damage to part of his frontal cortex. What happened is he's going to be working in an area that's clearing land for the railroad. And so as they were clearing the land, some explosives went off. And this pipe or this rod went flying through the air, and it actually punctured in underneath Phineas's cheekbone and went up through his skull. So it became clear that something in Gage's personality, once he had healed from this, had actually changed. And they think it's because it went through this section of his brain, and it was a result from the injury. And so it changed his personality completely. He used to be extremely nice, extremely hardworking, and he basically turned into what they refer to as a jerk after this. He wasn't very nice. Okay, here's some different regions. They have it colored in so you see the different parts of the cerebral cortex. We've talked about them already, like with the frontal lobe. We talked about it regulating speech production, motor control, smell, problem solving, and other sensory aspects or personalities. Temporal lobe is going to perceive and process auditory and visual sensations. It's going to be important in pattern recognition and language comprehension. The parietal lobe, which is going to be in purple, is going to receive and perceive touch and pressure sensations and important to sensory integration. And the occipital lobe, which is found at the back in blue, is going to be important in receiving and processing visual information. The corpus callosum is going to be divided into a left and right hemisphere, and it's going to be connected by a broad, thick band of neurons. So left brain versus right brain. The left hemisphere is going to be the side area that specializes in things like language, logic, and mathematical skills. These would be the type A personalities, they say, are more left brain. The right brain hemisphere is going to be more commonly known as the creative or intuitive side, the artistic and emotional side of the brain. So these are going to be the people that they refer to as type B or the left brain people. The limbic system is going to be within the forebrain. It's going to be near the center of the brain, and it's a set of structures that make up what's referred to as limbic. And so it's going to contain the cerebral cortex and two structures that are deeper within the center of the brain, which are going to be the hippocampus and the amygdala. And it's going to be responsible for many of our physiological drives and instincts. So we've determined a way to figure out how the brain allocates tasks. And so what we're going to do is we've learned how to measure brain activity. And they're going to do this through brain imaging technologies. So they're going to capture pictures of the brain as it functions, and it shows how those activity patterns work. So for example, a PET scan is going to be a positron emission tomography. The fMRI scanning is going to be a functional magnetic resonance imaging. And so here you're looking at a PET scan. Basically, they put in a radioactive glucose injection so they could see the differences. So the radioactivity can be traced in the brain as a subject performs a specific task. And because the areas with more metabolic activity require more fuel, more of the radioactive glucose is going to move to that area. So it shows the areas that are going to be active. And so looking at the left, you see a healthy individual. The brain is working hard. If you look to the right, that's going to be the brain of somebody that has Alzheimer's disease. The fMRI scan is going to be basically a giant magnet, and it's going to help detect relative amounts of oxygenated blood and deoxygenated blood in various parts of the brain. So when parts of the brain are going to be active, more blood's going to flow to them, and the magnet can pick up on that change. Interesting discoveries that we found from measuring brain activity is going to be seeing your favorite foods, for example. It's going to activate reward centers in your brain. So altruistic behavior and activities are going to be seen in reward centers. People with Alzheimer's disease exhibit significantly reduced brain activity, and they don't actually pick up on their favorite foods like they would used to. Specific brain areas are going to be involved in processes of things like learning, language, and memory. Language is simply the means of communication between individuals of any species. So it's going to involve two areas of the brain. You have the Broca's area and the Wernicke's area. In that Broca's area, it's going to coordinate the muscles necessary for speech production. In the Wernicke's area, it's going to be responsible for the comprehension part. So one part is responsible for the muscles for speech, the other is going to be comprehension. In 1999, an American woman named Tiffany Roberts had a stroke. Upon recovery, she had developed a British accent. And so with this British accent, it appeared that parts of her language production areas had been damaged by the stroke. So a reduced amount of oxygen to those parts of the brain actually changed her accent. Memory in the amygdala is going to seem to associate with emotional feelings and sensory input with those memories. The hippocampus is going to be important also because it's going to help with storage and retrieval of memories, as well as the transfer of short-term memories. Each fall, red squirrels are going to hide more than 3,000 acorns and other nuts. But when it comes winter, they're able to remember the location of about 80% of those nuts and recover that as a food source. So that's going to actually help them survive. So the better their memory, the more chance they'll have for survival through the winter. Drugs, unfortunately, can be a bad thing or they could be a good thing. Drugs, whether recreational or therapeutic, are going to be found in nature and made in a laboratory, so they can work to mimic neurotransmitters. So basically what they're going to do is they're going to trick the brain's signaling system. Some examples of different drugs that can do this are going to be cocaine, Prozac and Zoloft, morphine and heroin, and nicotine. It's going to occur in 4 steps, how it kind of tricks the brain or how it mimics a neurotransmitter. First, the drug's going to be taken, it's going to cross the blood-brain barrier, and it's going to reach the brain's pleasure centers. Then the drug molecule is going to bind to sites of presynaptic membranes where the neurotransmitters are normally going to be reabsorbed. As the drug molecules block reuptake sites, previous release neurotransmitters are going to remain in that synaptic cleft. That neurotransmitter repeatedly stimulates postsynaptic cells, causing nonstop activity in that brain's pleasure center. And so with the cocaine, it's actually going to do this and affect dopamine reuptake. Prozac and Zoloft are going to affect serotonin reuptake. It's going to be known as a selective serotonin reuptake inhibitor. Morphine and heroin are going to mimic endorphins. So endorphins are going to be natural painkillers that are found in the body. They result in a state of euphoria, so they make you happy. Nicotine is going to be the most commonly used drug. It's going to mimic acetylcholine, and it's going to cause cells to release adrenaline and dopamine. Do human drug addictions and dependencies reflect differences in our genes? Well, actually, they do. The allele of the D4DR gene is going to be found among 1/3 of smokers in a study, and it's going to be the same one that appears to cause individuals to exhibit a variety of personality traits that are going to be associated with risk-taking and novelty-seeking behaviors. So if you have this gene, you tend to be more likely, like this person here going bungee jumping, but you'll be more likely to possibly smoke or more likely to create an addiction to something such as alcohol. A brain slows down when it's in need of sleep. Caffeine seems to wake it up. But what is it actually doing? Well, worldwide, more caffeine-containing tea is going to be consumed every day than any other beverage other than water. A close second is going to be coffee. Now, coffee's my favorite one. With this caffeine, it's going to be found in both. It's going to interfere with the normal neuron functioning. How it works is the adenosine in the brain. The adenosine basically is going to bind to the receptors on nearby cells. Adenosine is a cellular waste product that's going to build up in a synapse. So as that adenosine receptors fill up with adenosine, then the brain neurons are going to become less likely to fire and perceived as tiredness. So caffeine's going to come in to the brain, and when you ingest it, it's going to circulate around in the brain and bind to the adenosine receptors. The adenosine receptors, since they're bound to caffeine, aren't going to be able to reduce the neuron firing, and the result is that you're going to be more awake. So basically, it's going to block those receptors that let you know, hey, I'm getting tired, I have a buildup of adenosine. Blocks them, you don't realize that, and you seem to be more alert, more awake. So caffeine seems safe for most people, and there's going to be no clear evidence that moderate consumption of caffeine increases a risk of much of anything, except for they have found one. They found that with a lot of these energy drinks and extreme high amounts of caffeine, not just what you're taking in through coffee, but the large amounts that are coming in through these energy drinks and through energy shots are actually increasing the chance of people having strokes and heart attacks. We're starting to see them as early as late teens, if they start to consume these around 16, 17 years of age, or even in 20s and 30-year-old people. So that is one thing that's kind of very scary that we're seeing going on. And so here's a picture of it coming in. The adenosine usually binds to the receptors, lets it know, hey, you're sleepy. But instead you have the caffeine come in, binds to the receptors, blocks it. You don't pick up that you're tired. Alcohol is going to be another chemical that can come in and interfere with many different neurotransmitters. It's going to be a great neurotransmitter impersonator, and so it can fool at least 4 main things in the body. It blocks glutamate receptors, so it results in a slow of reaction times and slurred speech. It blocks dopamine reuptake, and so it results in kind of a pleasant buzz or happiness. It's going to stimulate the release of endorphins, which results in blocking pain messages, and increases the efficiency of serotonin receptors. So that's going to result in an increase in contentment and happiness. And so by blocking receptors, it's going to be able to make you think that you're not in pain. It might make you think that you're happy. It's going to actually slow your speech down. It can act like cocaine, but it's going to be much weaker when it blocks the dopamine reuptake. And it can resemble morphine and heroin in respect as it greatly reduces the magnitude. Alcohol is going to spur your body to produce little opiate-like high. And like Prozac, alcohol is going to modify and increase the efficiency of our serotonin receptors. And so it increases the contentment that accompanies serotonin release at the synapses of the brain. So for most people, moderate alcohol consumption is going to be pleasant and doesn't have a high significant to health risks. For some, however, alcohol abuse or alcoholism could lead to serious problems. You could end up with cirrhosis of the liver, and it could be deadly. So long-term side effects, these include increasing the risk of some types of cancers. It increases the risk of liver disease, increases the likelihood of harm to a fetus during pregnancy. And so that's going to end our section on the brain and on our nervous and motor systems.